Can I start over? That was bad.
Yeah,
I lost where I was.
No, you're good. You're good.
I'm like, where am I? Hi, my name is Dr. Payne, and today we're going to be going over a couple fun questions for you. All right, our first question today is, how do DNA and RNA differ, and what are the implications that these differences have for molecular biology research? So, DNA is the genetic material found within the cell. In eukaryotic organisms like ourselves, that DNA is found within our nucleus, and it encodes all the information that we need to function as an organism. DNA itself is double-stranded, and it's protected in that place I just mentioned, the nucleus. RNA is the functional component of DNA. So the DNA inside the nucleus is transcribed into RNA. RNA leaves the nucleus, goes into the cytoplasm, and it's translated by these organelles called ribosomes. Once translated, we've created something called proteins, and proteins are really cool because they act by doing all the functional things within the cell. So why does this matter for molecular research? Well, scientists often want to know how cells respond to different environments. So if we want to know why a cell is doing something specific, and we're saying doing something, usually the researcher is looking at what types of proteins are being created in response to some external stimuli. So to analyze that, we can look at the RNA. In other words, what is the cell making in response? So if proteins are made, we know RNA must be out in the cytoplasm being made into proteins, and therefore what we would look at to determine what was causing those proteins to be made is actually the RNA. Our next question is about gene regulation. So understanding how gene regulation plays a crucial role in cellular processes. So genes are found in DNA. So that sequence of nucleotides that tells our cells how to function and provide all the information for life, that includes these small sections or sequences of DNA that we call genes. Gene regulation is simply how we either upregulate or create more copies of that genetic information, or downregulate where we're not making copies of that genetic information so that it can be created, made into proteins to act within the cell. So as the cell responds to different outside stimuli or sometimes internal stimuli, the cell goes, I might need more of this protein, or I need less of this. So by upregulating or creating more of something, or downregulating by creating less of something, the cell can maintain a level of balance or homeostasis throughout its life. So our next question is, what are the key principles behind DNA sequencing techniques, and how have they evolved over the years? So just as a reminder, DNA is a sequence of A's, T's, C's, and G's, but this sequence provides the genetic information for everything that allows a cell and organism to survive. Oftentimes, researchers want to know the very specific details of those A's, T's, C's, and G's that make up certain sequences so that they can understand what genes or specific sequences are being expressed by a cell. Initially, this was done by something called Sanger sequencing. So we can, in a test tube, actually make replications of DNA. And what scientists have discovered is that if they modify the different nucleotides so that they cause the sequence to stop extending, they can make short little truncated pieces of DNA. And if they run this DNA through something called an agarose gel, through a process called gel electrophoresis, they can separate out the different segments of DNA by length, with the shortest being all the way at the bottom and the longest being at the top. And what they can do is, based on which section of the gel of the 4 sections they load, one for A, one for T, one for C, and one for G, they can read the sequence A, T, C, G as it moves by length up that gel, and essentially end up with the entire sequence of DNA. Now, as you can imagine, this is quite time consuming, takes many, many test tubes doing DNA replication, and is honestly quite expensive. So in more recent years, The development of something called next-gen sequencing has actually allowed us to do this so much more quickly and more efficiently, to the point now that we've fully sequenced entire genomes of many organisms. So for this question, we have, what ethical considerations should researchers in biology education research keep in mind when working on projects involving student performance and access to classroom interventions? So as someone who received a PhD in biology education research, this specifically pertains to a lot of the research that I conducted as a graduate student and that I still conduct now at the University of California, Irvine. I really like this question because I think it really highlights some of the misconceptions when it comes to how research is conducted in the sciences. So most of us know that when doing a research project, something known as a control or a set of standards that are not manipulated in a study is really always done. However, in certain circumstances, such as education research, we sometimes can't ethically include controls. So this question really gets at that. So one of the biggest ethical considerations in education is when an instructor or when a researcher wants to put in an intervention, which is just a fancy way of saying we want to change something about the material or the classroom dynamic to improve student success, whether that is success in outcome or self-efficacy or even just retention at the school or their ability to graduate with a high GPA, we have to consider whether or not it is okay to only provide that intervention or that change to one subset of students and not to another subset. So when you think about this, if my goal as an instructor is to increase the way my students perform, increase my students' success, is it okay for me to consider to only giving half my students access to that particular intervention? And by most standards, that is actually considered not okay. So ethically speaking, I can't give half of my class this access. So there are two ways of mitigating this as an instructor or as a researcher in the field. Either collecting data from two separate samples of students. So for example, maybe I would collect data from this year's students over not putting in the intervention, and then collecting data from next year's students with the intervention. Obviously, there's a lot of caveats to doing that because I'm not looking at students from the same year, but it's more ethically acceptable because I'm not denying access to a full half group of students. The other option, and this is honestly the option that is taken by a lot of researchers, is simply to not have a control group and explain your rationale for not including that control, which in this case is simply it wasn't ethical to provide this to not provide access for half of my students to this intervention that is designed to increase their success.